Hello, my name is Ian Angstrom. I'm an undergrad at the University of Washington, and I work in the Sullivan Lab at the Fred Hutch. Our lab focuses on the metabolic constraints of cell proliferation and how to target those constraints in cancer. To start off with, cancer cells need to grow a lot, and to support that increased growth, they have a modified metabolism. One of the important molecules in that metabolism is the amino acid aspartate. Aspartate is used in the synthesis of nucleotides for DNA replication. It is incorporated into proteins, and it is used to create other amino acids. The amount of aspartate that cancer cells have available to them directly impacts their growth. The gene asparagine synthetase, or ASNS, converts aspartate into asparagine, effectively depleting that aspartate pool. By expressing ASNS to varying degrees and treating with drugs that prevent the synthesis of aspartate, we hope to find the types of cancers that will be most susceptible to aspartate suppression therapies. First, we generated a stable 143B osteosarcoma cell line that overexpressed ASNS and one that expressed EGFP as a control. We then treated them with increasing doses of rotenone. Rotenone inhibits the electron transport chain, and NADH then cannot be oxidized to NAD+. NAD+ is an important cofactor in reactions, like the synthesis of aspartate. So, rotenone can be seen as an aspartate-suppressing drug. We then gave some of the cells 1 millimolar asparagine in their media. We saw that control cells died at lower concentrations of rotenone and did not show increased resistance to rotenone when treated with asparagine. ASNS overexpressing cells survived at higher doses of rotenone, but more importantly, their growth improved significantly when treated with asparagine. In conclusion, we did not expect to see control cells dying off at lower rotenone concentrations, but those cells did grow slower, most likely due to the nature of single-cell cloning. The large gap in ED50 of the asparagine synthetase cells indicates that their growth can be rescued by asparagine at lower concentrations of rotenone, which is consistent with the expectation that because they are using up more aspartate to make asparagine, adding asparagine back to the cells will boost aspartate levels and proliferation rates. The next step will be running this experiment again, but using an inducible system for activating ASNS in cells. I'd like to give a huge thank you to Dr. Lucas Sullivan, Christian Davidson, and the rest of the Sullivan Lab for all their help, support, and patience. I would also like to thank Dr. and Mrs. Levinson for their generous support of my research through the Levinson Emerging Scholars Award.